Welcome to the first of this season's WALS lectures, and today is a special occasion. It is the William E. Paul lecture, and I'm going to start with, this is the professional picture of Bill, but I prefer that picture of Bill because I think all of us that knew him well remember him much more in this ebullient mode than in the more somber picture that preceded this. And I think the title here is correct. He was the immunologist's immunologist. And he even recounted in his brief autobiography, Endless Fascination, how he came to choose immunology. He read a small volume of essays by Michael Heidelberger and decided on a Brooklyn subway that he would become an immunologist. He then unfortunately deviated from this path very briefly when he was at NIH from 62 to 64 to do endocrinology, but then he saw the light, joined Benacer's lab in New York, and then moved with Baruch to the NIH. And following Baruch's move to Harvard in 1970, Bill became the chief of the Laboratory of Immunology, a post that he held until he passed away in 2015. So you can back calculate how young he was when he was appointed and how long he held that post and how wonderful the laboratory was during that time period. Now, for those of you who are not fully aware of it, he made many scientific contributions. The one that he is perhaps best recognized for is the discovery of BSF1, which is what you now know as interleukin-4. He really did an enormous amount of work unraveling how it contributed to IgE responses, how the signaling occurred, how it contributed to Th2 development. He was in large measure responsible for a classic paradigm in immunology now about the positive cytokine feedback regulation of polarization of CD4 T cells. He was involved in describing isotype switching that was mediated by particular cytokines. He discovered that mast cells and basophils also make these Th2 cytokines. And he spent a lot of time dealing with, if you want to call them, theoretical treatments of how self-tolerance is maintained in work that he did with Zvi Grossman. But this is a very abbreviated list of everything that Bill contributed scientifically. And I urge those of you who are not familiar with the rest of the list to go and look it up. Now, he made many other contributions outside of the laboratory with Dr. Fauci and Harold Varmus. He played an important role in the creation of the Vaccine Research Center, something that occurred when he was the first director of the Office of AIDS Research. He was on an enormous number of advisory panels. I still have a copy of his CV, and you have to wade through many, many pages to get through the list of all the ways that he helped other people in science, selection of honorific awards, on academic boards, and even on disease-focused funding organizations such as the Lupus Foundation. And then he also was an educator. Many of us know about him through his book, Fundamental Immunology. Maybe not everybody recognizes the fact that he was the initial editor and the editor for nearly 30 years of the Annual Review of Immunology, and during his tenure, it was the most cited publication in biomedicine, not immunology, biomedicine worldwide. He was a great mentor. You can quickly read this list and look at names like Charlie Janeway, Ron Schwartz, Laurie Glimsher, Mark Davis, Bob Cedar, Jeff Zuja, some people you might know from here, but many others over the course of his career. And I wrote a few words when he passed away that I still think ring true and is worth pointing out, but I'm not going to read all this and just paraphrase it, that science is made up of many little pieces that have to get woven together into a full tapestry. Some people contribute little threads. Other people are really the weavers of that tapestry, and Bill was a weaver. And I think that that's something that we all should keep in mind. And that also is a great segue to today's speaker, Max Cooper, because Max has many of those same qualities. He received his MD from Tulane. He then, for reasons that are unclear, decided to brave the northern wilds and went to Michigan briefly, then returned to the South. He then became a little peripatetic and then went to London and UCSF and then to Minnesota, which he assures me is even colder than Michigan. And there he worked with Bob Good and began to do the groundbreaking work that led to this year's Lasker Award in dissecting what at that time was a single small population of relatively nondescript cells called lymphocytes into two major subsets, the B and the T cells, and how they interacted and also subsumed different roles in the adaptive immune system. He spent 40 years on the faculty at University of Alabama, Birmingham, where he really did enormous amounts of work studying humoral immunity, discovering isotype switching, and with Owens and Raff, the bone marrow origin of mammalian B cells. He also did a lot of translational work using studies on the ontogeny of B cells to inform studies of childhood leukemia. For the last 15 years, he's worked on the topic he will address today, and rather than going through the longer description of that, because I always wonder why introducers give away what the actual lecturer is going to say, I won't discuss it anymore, except to say if you don't know about it, we'll be glad you're here today to hear about it. Max has won many awards besides this year's Lasker Award. He's a member of the National Academy of Sciences and Academy of Medicine and the Academy of Arts and Sciences and the French Academy of Science, and I'm going to run out of breath reading the rest of this list. He's been the president of many learned societies, including the American Association of Immunologists, and won a number of other awards, most recently before the Lasker, the Japan Prize. And so I just have one official announcement that I need to make. This seminar was sponsored by the Immunology Interest Group, and the FAS is sponsoring a small reception afterwards to which everyone is invited. And then, without further ado, our speaker, Dr. Max Cooper.
Thank you, Ron, for that gracious introduction. And I'm honored to be here to give the William Paul lecture. Especially honored that Marilyn, you would attend. Thank you so much. Bill was an immunologist, and he was a great friend of so many of us. He taught all of us a lot. He certainly taught me many things. I never published with him, but we did do actually experiments together at one time. He brought his student Joe Davey, who was at the top of that list Ron just showed, they knew how to do autoradiography, and we were trying to find out how frequent were B cells that would bind a particular antigen. And we were right on the nose for 1 in 10 to the 5th at that point. We never published the data, actually, and I forget the reason why, but we were, Bill and I enjoyed a lot of friendship together, and he certainly was very supportive of me and my career in many different ways. I'm really honored to be able to give this lecture in his honor. Well, I'm going to go back and try to trace the origin of our adaptive immune system. So the question is, how did our adaptive immune system arise? And we all came from the sea, it's thought, but how is it that we got the development of the kind of adaptive immune system that we have? I started this question to try and seek the answer to this question with Jan Klein, who was interested in evolution, was a famous MHC class, major histocompatibility class gene scientist, and we agreed to go back and start together to work on trying to seek the origin of our adaptive immune system. We knew at the time that all jawed vertebrates, all the way back to sharks, skates, and rays, the cartilaginous fishes, had basically the same kind of immune system that we have. They had a thymus where they generated T cells. They generated B cells in their bone marrow or hematopoietic tissues. They had major histocompatibility genes, class 1, class 2. They had RAG1, RAG2, recombinase activating genes 1 and 2 that were involved in the assembly of T and B cell receptors. And they all had B cell receptors, alpha, beta, gamma, delta, T cell lineage receptors, as shown by many investigators working in how the evolution of adaptive immunity in jawed vertebrates. But we couldn't go back and look at earlier jawed vertebrates because we knew them only through their fossil remains. They had all died out. It was thought somewhere around 360 or million years ago. But there were jawless vertebrates, lampreys and hagfish, that were thought to be even more ancestral or basal than vertebrates than jawed vertebrates. So Jan Klein and I set out to work together to study these jawless vertebrates. And the species that we picked were these sea lampreys, Petromyzon marinus. They are one of about 40-something species of lampreys that live worldwide in distribution. About half of them are predators, like the sea lamprey, and they spend a short time in their adult life, about a year or a year and a half, as predators. When they came into the Great Lakes of North America, they wiped out the fish. They have a very rasp-like tongue, and they have a ring of sharp teeth, and they latch onto the sides of these fish prey, and they derive their nutrients from the blood and tissue fluids. And one of these lampreys, it's estimated, can kill about 40 kilograms of fish in a year. So they really did wipe out the native fish when they made their way from the Atlantic Sea through canal connections into the Great Lakes of North America. They mate, and then the life cycle starts over with fertilized eggs. The larvae live for 3 or more years in these small streams that flow into the ocean, or Great Lakes in this case, before they undergo metamorphosis, get larger, retract the hoods of their eyes so they can see their prey, and the life cycle goes this way. After the adults mate, they die, so that it's a complicated lifestyle. If you cut one of those larvae, which is what we study mostly because it's difficult to get and keep the adults, you see something like this. There's around this very simple intestine is a tissue, a hematopoietic tissue called the typhosol. Some people call it the lamprey spleen. And Jan Klein and his colleagues had found a gene, a transcription factor, SPI-B, a member of PU.1 family, that is known to be important in hematopoiesis and B lymphocyte development in particular. They showed that it was expressed by in situ hybridization by a small round cell, and raised the question, could this be a lymphocyte? And so Jan Klein and I set out together. He was funded at that time by the Max Planck Institute, and in addition to NIH support, I had Howard Hughes support, and that allowed us to go back on this fishing expedition. I thought that we could isolate those cells that might be lymphocytes by their light scatter characteristics, just by forward and side scatter light reflection. And indeed, there was a small button of cells that looked like intestinal epithelial lymphocytes, physical structure or light scatter characteristics. When we sorted them, they looked like small lymphocytes. Even by electron microscopy, they looked just like our lymphocytes, except they were a little smaller. But that's a very indescript cell picture. All it means is that most of the cell is nucleus, And in the cytoplasm, which is sort of scant, there are not many organelles there. So we made cDNA libraries to try and get sequences of genes that they were expressing to see if they expressed the genes that our lymphocytes express. And when we got some 2,000 sequences, which was quite a lot just in the early part of this millennium, as we converted the sequences into amino acids and then annotated our catch, we couldn't find any of these cardinal elements of our adaptive immune system, although we found lots of genes being expressed that our lymphocytes express for differentiation, for migration, and for other essential functions. We went back, actually, Zev Panzer, who was a molecular biologist, marine biologist, joined my laboratory at this time, and Zev and I decided, after trying again and with the same result, to see if we could catch cells in the act of responding. Others had shown, including colleagues Bob Good, whom Ron mentioned, who was my mentor, had shown many years earlier that these creatures could respond, these lampreys, could make agglutinins, things in their serum that would clump an antigen like a red blood cell, a human O-positive red blood cell, in a specific fashion, but no one could find evidence for an immunoglobulin or what the basis of this was, and that result had sort of been discredited. But Zev and I decided that we would go back again. This time, we would stimulate the lamprey larvae and then see if we could catch cells responding to the immunization in the act, so to speak. And so we injected lamprey larvae with live bacteria, E. coli, sheep red blood cells, and 2 plant mitogens that stimulate lymphocytes in jawed vertebrates, PHA and pokeweed mitogen. And after that, we saw expansion of a population of cells that by light scatter were larger, and when we isolated those, they looked exactly like lymphoblasts our lymphoblasts that respond to stimulants such as these. So we made a differential library, cDNA library from this population of blasts, and then got sequencing, and then started annotating our catch. And we ran into we saw none of the cardinal elements of our adaptive immune system, but we saw lots of leucine-rich repeats, which we didn't think was so interesting, because leucine-rich repeats are used by everything on our planet. Toll-like receptors are a good example of leucine-rich repeats. And in sea urchins, there are hundreds of genes that encode toll-like receptor sequences. But in the end, we decided a summer student from University of Michigan wished to learn some molecular biology techniques, and so we assigned her the task of sequencing these ESTs, express sequence tags. And at the end of the summer, she showed her sequences, and what hit us in the eye at that point was the fact that each one of them was different from the other. They had ends that were constant in sequence, invariant, but in the middle they had different numbers of these leucine-rich repeat segments, and the ones that they shared were all different from the other, as you can tell from this scorecard, the color coding. And finally, the light came on for us that although this was not what we started out looking for, perhaps this was the kind of variability that could give a recognition system that would allow discrimination between different antigens. Now, that longest sequence is depicted in a cartoon here and the sequence here. And interesting things about it is that the part that's highly variable in the others are these, each one of them had an N-terminal and C-terminal LRR region that were fairly lengthy, a shorter LRR1, and then these green blocks of all of 24 residues in themselves, plus a connective peptide, that were where the variability was occurring. Stalk region was interesting too in some ways. It was rich in threonines and prolines, suggesting that maybe it had flexibility. It had, at the carboxy terminal region, several cysteines, 8 in all, and I'll come back to those, but the interesting other signature that we could see was a sequence that suggested that they might be clipped at this site and expressed on cells by a GPI linkage instead of expressing all of the carboxy terminal sequence. We wondered just how variable these might be, and this is a phylogenetic cartoon that shows how closely related the first 112 VLR clones that we sequenced. And these came from 13 different larvae, and also we found later that they are the same from adults. And the way to look at this diagram is that these two are the closest to each other than any of the others, and they're 5, 10, 5 back up, 10% different in sequence from each other, and so on around this clock. There are a couple of points I'd like to make from this. There were only duplicates, or the same sequences were seen 3 times, and all of those came from the stimulated larvae that I just mentioned. And also this asterisk means that single cell PCR, polymerase chain reactions were done, and each one of them only expressed one sequence, implying that each of the cells that expressed them were monoallelic in their expression. So we wondered, of course, what's the genetic basis for all this diversity? Are there many genes like the toll-like receptors in sea urchins, or the plant-resistant genes in plants that are involved in defense? They encoded by each one by a different gene, or are there multiple? Is there some other basis? So at that time, we found by contacting Chris Amamiya in Seattle, who had made a genomic library of pieces of DNA from the sperm of a single lamprey. And we looked through his library, and we could only find one VLR gene, and it was very incomplete. It encoded for a signal peptide, the N-terminal region of LRR and the C-terminal region in 2 pieces, but none of the other blocks, the green or the light blue that I showed before. In between these pieces of the incomplete germline gene were large stretches of about 6 kb each of non-coding intervening sequence, and then the stalk region, the invariant stalk region. When we could get sequences in the flanking regions, and that was not so easy because the there are lots of repeats, and it's hard to get continuous sequences, which is still a problem for looking at the genome of these creatures, although it's much better at this point. But we found, flanking this incomplete germline gene, genes that, if brought in somehow and stitched into the germline gene, and deletion of the non-coding intervening sequence could give rise to this mature VLR gene that are those genes that we were encoding, finding in our analyses. So we isolated our semi-purified cells with lymphocyte characteristics, like the ones I showed earlier, and red blood cells, and then using these forward and reverse primers, we could find that the large germline gene product was present in both types of cells, but only in the lymphocyte population were these shorter, smaller products. And we sequenced many of these clones, and like the others that I mentioned, all of them were different in sequence, one from the other. So we started out with this model of our results. So we, by this time, we had over 600 different, we call them variable lymphocyte receptor genes, because we could show if we expressed a construct with a tag in mouse thymocytes, T cells, the product came to the surface that we could find with our tag, and also we could cleave them from the surface by phospholipase C, indicating that they had a GPI linkage to the cells that they were expressed by. So we call them variable lymphocyte receptors, or VLRs. We submitted over 600 different sequences to Igor Rogozin and Lax Ears in the NCBI, and they calculated, these computational biologists, that the potential repertoire for these for lamprey lymphocytes expressing these VLRs would be greater than 10 to the 14th power. And actually it turns out to be higher than that, because they were calculated on the basis of each one of these donor segments being complete, and they're given in small fragments. But that's equivalent to the potential repertoire of our B cells in the light and heavy chain assemblies, about 10 to the 14th in repertoire diversity. We assumed that they would probably be secreted or released in some way as multimers in order to explain the agglutinins, like the ones that agglutinated O-positive human red blood cells in an apparent specific way. We set out to test this hypothesis by immunizing lamprey larvae, and here you get an idea of the size. They're about the size of a small pencil, about 12 centimeters. And this is an insulin syringe. You can see the fingers of the immunizer. And we inject them into their coelomic cavity, which is equivalent to our peritoneal cavity, our abdominal cavity. You can only inject a small amount because it leaks out if you give more. And we immunized them, and then looked 2 weeks later, all of them made agglutinins in a fairly low titer of about 1 to 100 on average. But if we gave a booster immunization, we got a 20-fold increase in the level of these agglutinins. We made a mouse monoclonal antibody that was specific for that invariant stalk region. And if we absorbed this serum with that antibody, we could remove all of this agglutinin activity. So it seemed clear that these agglutinins were the actual proteins that were the variable lymphocyte receptors that were causing the agglutination. If we use that antibody with a green fluorescent tag to identify cells that were expressing, we call them VLRB, and with a red tag that was incorporated by cells that were synthesizing DNA, and a blue tag just to light up any nucleated cell, you could see that in this, these are cross sections of gill folds. They're cut crosswise. And in these small vessels, you can see there are lots of these VLRB producing cells. And this is before and after immunization with the anthrax exospores. And you could see that cells that were dividing and making these green VLRB proteins were increased in number after immunization. The cells, though, that were actually secreting the agglutinating activity were not these lymphocytes, either the small or larger ones, but came from the cells that had these light scatter characteristics, indicating larger cells that expressed the, on their surface VLRB that Matt Alder, an MD PhD student who did the experiment, could show by an ELISpot assay. These were the ones that were secreting the antibodies. Now, these are plasmablasts, and just like our plasmablasts. They also have mature plasma cells with much more manufacturing channels of ribosomal laden channels that are typified plasma cells. So they have plasma cells that are identical to, in appearance, to ours, but we wouldn't detect them because at this point, at the point of plasma cell differentiation, they would have lost their surface VLR. Brant Heron, a postdoctoral in the lab, did a Western blot using this stalk region antibody that sees all of these VLRBs regardless of their leucine-rich repeat variability. And you could see that in the serum, they are very large proteins, much larger than any of our standards. But if he reduced the disulfide bonds, and remember those cysteines at the carboxy terminus, then he could reduce them to much smaller blots. You can see that maybe about 10 of these or more would be in the polymeric form of these VLRBs, each having one of these. You can also see that they're different in sizes at both treatments. And as you would expect, given the fact that they can have different numbers of these green blocks and therefore of different lengths. So this is not something that you could begin to do biochemical analysis. So Brant Heron designed a way to make monoclonal VLRB antibodies. And so he immunized lampreys, prepared RNA and constructed cDNA libraries that he expressed one by one in human embryonic kidney cells, and then plated these cells in 96-well plates, and then looked in the fluid that they were living in for expression of VLRBs and for antibody activity against the antigen. And in that way, he isolated a monoclonal antibody that was against a red blood cell O-positive human O-positive erythrocyte. And Ken Roos at Florida State University took this sample and did a negative image electron microscopy to give this kind of image, and here's the cartoon that interprets the image that can derive from Brandt's monoclonal antibody. And so there are 5 pairs of dimers that are joined by these disulfide bonds at the base of the stalk region. I mentioned that there were 8 of these cysteines that could create these disulfide bonds. At least 4 of them are required for secreting in this polymer, this multimer fashion. If Brandt truncated the constructs to not have the sequence for these, this carboxy tail, then they were released from the transfected cells in singlet forms. So this stalk region, if you remove this carboxy terminal that accounts for their being held together at the base, is the way that they're expressed. We still don't know exactly how this conversion from GPI linkage to the secretory stage by the mature plasma cells works. So this is a model that we had then. One of the questions that I had was, which came first, B cells or T cells? It was a question that I was often asked. And I thought it would be B cells, because this is a simpler way to make an antibody for protective purposes than making T cells, which have to have antigen-presenting cells, can only see antigens in pieces in the main. And so this would be an earlier way of making antibodies in an evolutionary sense. Actually, that turned out, as most of my guesses, wrong. Again, we had found in hagfish, which is a sister group of jawless vertebrates, 2 of these VLR genes, which were also flanked by multiple sequences, incomplete germline genes flanked by segments that could be brought in to generate diversity. And later, Kasahara identified, who works in Sapporo in Hokkaido, found a 3rd one, and we found that in hagfish as well. So in both hagfish and lampreys, there are 3 of these germline genes, all of them incomplete, mainly having bits and pieces missing in the middle, the variable portions of these genes, but flanking them hundreds of these potential donors that could be brought in bit by bit and then assembled using a short stretch of similarity or homology between the donor and the recipient to bring in first one like this. It has the dark blue with a matching sequence held in place for a while, and this gets copied in, and then to get from there, one has to have one that has these green sequences, and so on down to the end, and deleting at the same time these non-coding intervening sequences. And a group from Tokyo, Nakata, and his colleagues showed that this could start on either end from the 5' end and go through to completion, or from the 3' end and go back the other direction to fashion a completely assembled VLRB gene. But it only happens on one allele and only starts in one end or the other, never both at the same time. And then using monoclonal antibodies that would discriminate between these invariant sequences of the VLR-A, B, and C type of cells that express those genes, we made monoclonal antibodies, Masa Hirano and Fungoa and others in the laboratory, that could discriminate 3 populations of lymphocytes. And our data suggests that they are all derived from a lymphoid progenitor. And in a thymus equivalent, at the tips of those gill filaments that I showed you cross-sections of before, there are sites where genes are expressed that are normally required or have been shown to be required for T-cell differentiation in mice, and also in some cases by genetic defects in humans as well, to give rise to two prototypic lineages of T-like cells in the case of the lamprey, except that these are not GPI linked. They are transmembrane receptors. They are not secreted as the VLRB genes are secreted by this B cell-like. And again, Rogozin and Zev Panzer, and then in his own lab, discovered two genes that are like activation-induced deaminase, which is essential for the generation of diversity of B cells by class switching and by affinity maturation, and also for gene conversion that birds use for generating diversity of B cells. But in the thymus equivalent is another, the second of the cytidine deaminases that are like AID in us, that's expressed, and we could show that it's only expressed in this thymoid or thymus equivalent region, and then they are assembled first in the order of VLRC and then VLRA, as our data suggests. This being more like gamma delta T cells, one of the T cell lineages in our that we have that we still need lack complete evidence of what it actually is doing and what it sees. And then the other, the VLRA, like our alpha beta population of T cells, about which we know much more, and about which that differentiate into multiple different helper, inhibitory, all different kinds of T lineage cells. We have data that's suggestive that they are expressing cytokines, in this case like interleukin 17, that sees a receptor on the B cells. It's made by the VLRA cells that suggests they have cooperative interactions through the cytokines and cytokine receptors that they express. But really, we know not so much yet about exactly what they do. So here's the expression patterns that led to that conclusion, or some of the data that led to that conclusion. So if you isolate VLRA cells, VLRC cells, they express genes that are involved in T cell differentiation, much like our alpha, beta, and gamma delta T cells. Whereas there's some genes that are differentially expressed by one or the other. These expressed in particular by a gamma delta lineage of T cells in mice and humans, and these expressed more preferentially in alpha beta type T cells, and some they share in expression. The VLRBs express genes that are much lookalikes to cells like the CD8-2 that I mentioned, and other genes that we know to be essential in B cell differentiation. Here's an exception, a CD4-like gene. It would normally be expressed by alpha beta T cells in us, but in this case, in the lampreys, it's expressed in B-like cells. So 3 different lineages of cells, T-like cells that don't secrete their products, that can respond to proliferation when they see when they're immunized with other other lampreys that are genetically different from them because they're outbred. These are the two genes that I mentioned. The AID-like is a multi-exon gene. CDA1 is an intronless gene, and this is the one that's expressed during T-cell the VLR-A and C gene assembly on T-like cells. This one has other transcripts that are not shown here because part of the gene is missing in this initial report, but with the longer sequences here, these all have DNA expressivity or function, and they're all mutagenic. These in particular are highly so. So this is the current model, and I'd like to spend the rest of the time discussing more about the antibody products that these VLRs produce. But first, what do we conclude from this data? Well, first of all, we conclude that the genetic program for these two prototypic T-like lineages in lampreys and hagfish and a B-like lineage that are very similar to the genetic program for our T and B cells. Those genes must have existed in a common ancestor. The last time we had a common ancestor is estimated to be a little over 500 million years ago. So this program was already present by then, before the convergent evolution of different kinds of receptors that generate a diverse repertoire of antigen receptors in jawless and jawed vertebrates. What they were using before then is one of the many unanswered questions. Also, it suggests that the AID, or cytidine deaminase equivalent in jawless vertebrates, the gene for it was already present in both an ancestor of jawless and jawless vertebrates. But as I mentioned earlier, the jawless vertebrates don't have any of these cardinal elements that our immune system is used to make antigen receptors for T and B-like cells. Instead, they have these VLR-A, B, and C genes. So we reckon that this is two different solutions or convergent evolution to the puzzle of trying to create a diverse repertoire which will allow for memory and specificity in reactivity of the T and B cell lineages. So what about these B cell receptors that are secreted, these lamprey antibodies, if you wish? This first monoclonal that Brandt produced that was specific for O-positive erythrocytes, that's against a 3 saccharide combination, or H trisaccharide. And Byung-Woo Han in Ian Wilson's laboratory in Scripps was able to get crystals and solve the structure of this ligand or antigen interaction with this antigen binding site, and this cartoon shows in a ribbon diagram. So there's in this a hydrogen bond between a residue in this LRRV2. There are 2 hydrogen bonds in this N LRRV. And in addition, there are important interactions with the tryptophan between this H trisaccharide that's present on a loop that's encoded, that's highly variable, and encoded in this C-terminal portion of LRR that's highly variable. And in a space-filling model, it's easier to see. The gray is the H trisaccharide, And as you can see, it's sort of like the thumb that's sticking out here from the base of a hand, a curved palm of the hand, and with this one loop that also is binding, forms a pocket for this VLR antigen. So I haven't mentioned before what is inducing this highly variable region, and I'd like to just show you because it gives a little bit of an insight into how this huge variability actually can be generated of greater than 10 to the 14th or 15th or more. So here are we don't have continuous DNA sequence for the whole genome, and this is actually for a Japanese lamprey, and I've been telling you about work done mainly on the sea lamprey from the Atlantic Sea. And the Japanese lamprey has 2 of these extra bits in the middle of the germline gene, which is this configuration. And these are 2 large contigs or of DNA sequence that have been put together by Sabi Das, a computational biologist in our group. And you can see that there are multiple ones of these potential donors in these 1, 2, and 3 scaffolds from continuous sequences. So to start, if you started on this end, you would have to bring in one of these or sequences from one of these pieces that would be complementary by a short stretch of DNA sequence with the germline sequence in order to get to the next stage. And in order to move it forward, they would also have to have one of this sequence from this blue, which comes next. Then to get from the blue to the greens, you would have to have these donor sequences that had at least some of the blue and the green, or they could have more, as shown here. In this contig, there are none of them that could take it to the next stage, and nothing here either. For these green blocks, which are more numerous, there are lots of these potential donors in all of these contigs except for this shorter one where there are none. And you can see evidence of duplications to see one way of how they actually got to be so numerous as a mechanism for generating these donor sequences. To go all the way to here, one has to have a sequence that has this a little bit of green along with a connecting peptide and then a bit of this LRR C-terminal region. And you can see that there you find them in both of these, not in this short stretch. For the last bit to change here, you have to have these gene segments, and there are a few of them in this one, but in this little shorter one, there are multiple ones of these. And the junction of that last bit to here is what causes, this highly variable region in this C-terminal or LRR that forms this loop. So these would form the loop, and 85% of these all have coding regions for this highly variable loops, whereas these are shorter and they would not, 15% don't have the loop at all, but they still are made. So if you looked as a comparison of of a mouse antibody, the light chain and heavy chain variable regions, with an antigen, and here again, this is a trisaccharide, a carbohydrate antigen, and this is the lamprey antibody that I showed you earlier. So there are 3 loops for the light chain and 3 for the heavy chain that can have wiggle capability, conformational adaptability. So that's probably the reason that these regions which contact the antigens make antibodies of the kind that conventional antibodies that we and mice and rabbits and so forth make. They are highly promiscuous, or for example, you can take any monoclonal antibody, and if you screen for reactivity, you can find other antigens that it will bind, many of them having no detectable sequence identity. Whereas in these lamprey antibodies, this is the only part that has conformational adaptability. And that's probably the reason, this structural reason, we think is one of the reasons that the lamprey antibodies are so highly specific. In every comparison we've made with multiple clones or cloned antibodies from mice with those from lampreys, the lampreys are always more specific in their reactivity. Here's an example of their reactivity of this monoclonal that I showed you earlier with glycans or sugar determinants. These lampreys are particularly good at making antibodies to carbohydrate antigens. For these N-linked carbohydrates, 611 configurations of which are loaded onto this glass slide for screening purposes. And the monoclonal lamprey antibody that I've been showing you reacts with only certain ones of them and not at all with the others. And the ones that it sees all have a fucose as the first residue carbohydrate with an alpha-1,2 linkage to a galactose, which is linked in a 1-to-4 linkage to the next sugar down the line, which can vary one from the other. And we've made and screened by a more effective screening mechanism using yeast surface display, where we can screen millions of clones at one time, 2 other monoclonals that react with this H trisaccharide, and by changing that, one of which is more specific than anything that's ever been seen against the O-positive blood group trisaccharide. And by looking to see the differences in them, they're almost similar. They're about 80% similar in sequence. But picking out residues that you can change by structure-guided mutation, you can amplify and increase the specificity so that it no longer reacts with these carbohydrates, which are very similar, as does the parent one. So they are small. They have high affinities and low affinities, some of them. They can have functional effects, and they can recognize protein antigens as well as so this I won't go into in detail, but I just want to make one point. The little variable region that's encoded by this loop that comes out that I mentioned earlier is not always involved in binding to antigens. These are ones that have been seen before. This is the one that I've been talking about most. But these either don't have the loop for reasons that I alluded to earlier, or they don't use the loop even if they have it in binding to the antigen. So because of these advantages and easy engineering, because there's only one type of gene, we thought perhaps because of the phylogenetic distance and lack of tolerance constraints that we have, and that they might be able to see antigens that are normally ignored by our conventional antibodies. And we have 2 examples that I'd like to briefly show in closing that are proof of principles of this prediction. This is an antibody that Gertz Ehrhardt made by immunizing with multiple myeloma cells, the malignant plasma cells. And he selected a monoclonal lamprey antibody that only reacts with plasma cells, both human plasma cells and non-human primates as well. It doesn't see any of these early stages of B lineage development, nor of other cell types present in spleen, blood, tissue, tonsils. Here's another one that Gertz Ehrhardt made. And that one turns out to be specific for actually a well-known surface ectoenzyme called CD38. And that was totally surprising because CD38 is a cell surface protein with enzymatic activity that is expressed by all blood lineages early in their development, and even some subpopulations of T cells. So it was a great surprise to us that it was seeing this particular antigen. It turns out, as a Japanese group have shown, that this CD38 is expressed mainly on the surface of cells as a monomer. At higher levels, it forms a dimer. And at highest levels, it forms a dimer of dimers. And a carbohydrate addition at that point prevents further multimerization of this CD38 molecule. This MM3 recognizes the dimer of dimer forms. And it sees a site that is close to, or maybe is the site, where of its ectoenzyme activity, because NAD glycohydrolase activity, using a non-degradable substrate will block reactivity by this antibody, and vice versa. The antibody will block the binding or functional activity of this phospholipase, glycohydrolase, excuse me. This one is another one that he made by immunizing using plasma cell lines. And it sees only memory B cells and plasma cells, and not, it sees both memory B cells that still express IgD or not, but doesn't see the other types of cells. Here we're looking at blood and tonsils. This one turns out, to, even more surprisingly, to see a molecule, MHC class 1, that's present on every cell in our bodies, except red blood cells that have lost their nucleus. And this this antibody sees a special sulfated tyrosine on that class 1 molecule that's made by memory B cells and persists into the plasma cell stage. And if you delete one of the two enzymes that can add this sulfatase, then you lose the reactivity. So I'd like to close this by saying that many people have participated in this work. I mentioned many of them during the talk, and involve the work of many different colleagues and collaborators. I didn't mention enough Thomas Boone, with whom we've collaborated to define the thymus equivalent in lampreys and many other things in this model. And in closing, I'd like to acknowledge that I'm a co-founder of an early stage company, Novab, a contraction of novel antibodies that makes lamprey antibodies potentially for biomedical uses. And with that, thank you very much for your kind attention.
Thank you, Max. And although we are running a few minutes late, I'm sure Max would be happy to answer a couple of burning questions. So please use the microphone. Of course, Jay.
A beautiful series of studies. So even though these VLR antibodies see carbohydrates and have some hydrophilic residues in the combining sites, the fact that the framework is made of leucine-rich regions, does that affect the range of specificities at all, perhaps favoring hydrophobic antigens or antigens that have hydrophobic regions that can also interact with these framework leucine-rich regions?
I don't have a good answer to that question. We haven't noticed any constraints in that respect, but I couldn't say that there are none.
Max, great talk. Do you know anything about antigen presentation to these VLRA cells? Do they see native antigen or some form processed antigen? Or there's no MSC molecule, so who would? You see what I mean?
We know that the VLRA and VLRC cells, the T-like cells, respond with proliferation preferentially to allo stimulation. Whereas the VLRBs respond equally well to allo or any other antigen, protein or carbohydrates. But we don't know what the allo determinants are. That's obviously a key question, and therefore can't answer anything about antigen presentation and so forth.
So in the absence of identifiable RAG genes, what are the proteins and mechanisms that mediate the recombinase activity for the VLRs?
I'm not sure what, but they have lots of the same characteristics as the AID assembly or diversification of the gene conversion, diversification of avian antibodies. We have lots of, we don't know the basic biochemistry. I didn't point out clearly the donor sequences don't change location. They are never moved from their original location. They merely serve as templates for copying in the sequences. So, and these cytidine deaminases are incriminated to be involved as keys to that function, but not, it's a different mechanism than regular gene conversion where you change the sequence of something that's already there. These actually bring in sequences from the donor template, using it as a template. We do now have, Thomas Boone has unpublished data, and so do we, that's not quite as far advanced as his, that if you do a knockout using CRISPR-Cas system, you can prevent those knockouts lack the ability to express assembled VLRB genes. So they are essential, like AID, in a gene conversion-like mechanism, the molecular characteristics of which, or details of which, we still don't know.
Thank you.
I think we're going to have to move on, so let's thank Max again, and there will be the small reception in the library. Thank you. Thank you.